Now, I would like to welcome Dr. Shamir Khader. He would be talking on Pharma and AI, integration of Omics and Clinical Data. Dr. Shamir Khader is the Senior Director of Advanced Analytics, Data Science, and Bioinformatics at AstraZeneca, a global science-led biopharmaceuticals company. At AstraZeneca, he leads a global team of data scientists, biomedical scientists, statisticians, and software engineers as well. Over to you, Dr. Shamir.
Thank you. Thank you for the kind introduction. And thanks again, Elia, for hosting me today on this and it's Sunday, but I can already see that there are more than 100 attendees in the talk. So I know the science is strong here. Kudos to you and the team for organizing such a wonderful presentation, wonderful meeting. Let me just get this slide shared. Okay, can you see my slide?
Yep, we can see the slide. Great.
I just want to make sure I share the right one. Okay, it should be fine. Okay, you can see my slides, right?
Perfect. Thank you.
Okay. Yeah, thanks again. I'm here to talk about two of my favorite things, like about pharmaceutical industry in general, and also about AI. And then in a sense, what I'm going to do today is kind of give a little bit of a background about why this is important. Why is right now the moment for these two sectors to kind of come together, and with a specific example of how omics data, or for that matter, multi-omics data and clinical data that we typically generate, one will be generated in an experimental setting, whereas the other will be mostly generated in a clinical setting, how and why we need to put them together. So this is more of a pedagogical style talk. The reason is that Elia and the organizers kind of mentioned that there will be like a large number of students attending this meeting. So rather than brand new data, what I'm going to share is some of the published work, and then also show how we are developing some advanced analytic solutions to address some of the current challenges in integrating these types of data using a combination of machine learning and machine intelligence approach. So my group's research focus, we're primarily can be divided into sort of three main themes, like translational bioinformatics, computational medicine, and healthcare data science. But the group, my leader, machine learning research group, and the focus really is to leverage high-dimensional multi-scale data to design, develop, and repurpose new therapies. That's the single line of research focus. But how do we do this? We are a computational team. We are from the get-go leverage advanced machine learning approaches. What we really wanted to do is we really want to sort of close the gap between the way we generate data, the way we interpret data. For many of you who are on this call, I was very much familiar that the current era of biology, especially I would love to call it digital biology, is moving at a very fast scale and pace. But at the same time, if you go into a hospital, you'll be still measured your blood pressure using a blood pressure cuff that's, I don't know, 60 or 100 years old, or you're using a stethoscope that's like, 100 years old, older technology. But how can we bring these two, different worlds together? Maybe we could help here as a group of data scientists and machine learning scientists. We could help with the combination of data and technologies. And why data is important. And we might have heard about big data, some call it deep data, but for me, data at any scale or any grade, level, range of quality is interesting, whether it's big, deep, small, or data crumbs, doesn't matter. As you can see that these are some of the world's most popular magazines, all the way from Fortune to Wired to Popular Science to Economics, and then also two of the leading science journals. They all talk about data can bring in value. But I would tell you that it's not just the data, it's about what question you ask with this and what tools do you use to mine this data. And then we'll see some examples as we go forward today. And let's kind of come back from that, specifically to the setting in biology. Like for example, from if we want to go from a disease, for say for example, diabetes to a mechanism to a therapy we will bring it to the patient, and then a patient gets a better outcome. Can that drug be working for something else? And a good example would be like a classic example would be metformin, like that. But then what happened is that we're seeing a paradigm shift thanks to both the technology aspects as well as improvement in biological profiling technologies as well. So there are like both the scientific as well as the technology is in a paradigm shift here. So for example, we are moving from a traditional data type to evolving data type. You don't need to have all of your data in one location to do centralized analytics. You can now do decentralized analytics. You can go from GBs or TBs in data to petabytes and exabytes of data. Your data can be structured, can be semi-structured or unstructured. You can use NLP, text mining, and other types of techniques to bring structure to your data. The data model could be a step, in olden days, like I still remember, like when we do a health informatics project, we need a stable data model. That's gone. That time is gone now. Your data model could be evolving flat data model. Data is moving from a low dimensional to high dimensional, and statistical approaches, and hypothesis-based approaches are now kind of moving to more machine learning or deep learning based approaches. I'm not saying one is better than the other, but we can combine these two, and you can see this in studies. For example, let's take the same example of diabetes, type 2 diabetes. You can see that one of the first GWAS had maybe 1,000 patients, but the last GWAS on diabetes have like 100,000 plus patients in that study. So as you can see, we are in this paradigm shift here. But at the same time, we also changing our data source. Earlier, we used to either a lab will generate a data set, they will analyze that, they'll have an insight. Or maybe we collect studies, like 10 studies that are related, and we do a meta-analysis on that. We don't have to be restricted like that anymore. If you have a good question, you can bring in data from anywhere. But there are like data repositories that are out there. Obviously, some of them are a commercial product, but some of them, like UK Biobank, is completely available for or those who are interested to ask interesting research questions with that type of data. So you have biobanks, you have biorepositories, and then earlier in the days, it was very difficult to get clinical trial data to do reanalysis, but that is also changing. We now have several projects that tries to bring in clinical trial data in an open access way, for example, whether it's ct.gov or aggregate analysis of ct.gov. So you can see that all of that's coming together. And then not just this, now we can measure, like, typically, like, when we run a clinical trial or study, when a patient comes into the hospital, we can measure a few things. And then when they leave the hospital, we won't be able to measure anything anymore. But then that's not the case. We now have sensors and wearables, which are affordable and accessible and can be integrated with your study program. And that brings you a variety of data as well. So you just don't have to restrict to just the omic data that you typically measure in a lab setting. You don't have to restrict to a clinical data that you capture in a hospital setting. You can now get into the real world and collect data from there. So that's one of the gist of the changes that we are seeing right now. And this is why this is possible right now, because we see a huge shift in the technology landscape, and a lot of technologies are kind of growing together thanks to access to high performance computing, and whether it's through cloud or local or a hybrid approach, or advances in both the biology as well as technology, like I mentioned, whether it's better natural language processing approaches, like you can mine a large amount of data together. You have more intuitive data models available. You can have a precise integration, governance, and security to ensure that the data is being accessed in a privacy preserved manner. So these are all possible right now, thanks to all of that progress. But when we come to drug discovery, development, and repositioning, what's all possible? So this is just an example of what's possible if we bring in AI into the whole enterprise of developing drugs. So it can definitely help in novel therapeutic agent discovery. You can do precision disease stratification, for example, within a population who are the disease population. Within that disease population, what's the subset of patients that need treatment A versus treatment B? You can do integration of multi-omic data, which we will see today. And then we can also use the same type of technology to understand that, how physicians are utilizing the therapies that we develop. When, like I mentioned, when a patient is not within a hospital, not enrolled in a trial, we can still monitor them if they're, using an approach that's called continuous remote monitoring and diagnostics. And then also we can plan, design, and optimize our trials as, using AI-based approaches. So just to kind of give a broad overview that AI is being applied across this enterprise of drug development. But due to the time constraints, we'll only focus today on the integration of multi-omic data, and we see a few examples on that. And even within this particular focus area, as you can see that there are a lot of possibilities here. But we'll pick 3 examples, like I mentioned. The first 2 are published one, and the last one, it's an ongoing project. I want to share, give you an early view into the project, and thanks to our leadership, this is currently being developed as an open access project. We'll be talking more and more about these projects, like these type of community activities, and we'll be happy to share the tool with you all, and would be great to hear from you about the tool in hopefully before end of this fall. That's our plan. Okay, before, so that's sort of a background of my talk. So I'm going to talk about 3 examples today. This is my first example. For those who are new to the whole genomics, clinical genomics-based drug discovery, this is a quick example. You might have heard about loss of function variants. These are genetic variants that could lead to an aberrant RNA product. And what happens is that you will end up with a non-functional protein product. And the concept is there in the literature. We have seen that it's been successfully used to find targets and now being translated to therapies across the industry, that you can see. And then the key 3 approach that loss of function could enable is you can probe biology. You can if there is a loss of function that leads to variant that leads to let's say loss of a functional protein product, would that lead to a particular phenotype? What is its clinical implication? Can we use that approach to develop a therapy? These are the things we can do with the loss of function approach. And there's a lot of research has happened over the last few years. But what we were interested, this is a work that I've done in collaboration with my colleagues from Mount Sinai. The reason was that what we wanted to know was, like, let's say if we have hospital-based clinical data, and then also have a biobank that's linked with clinical and genomic data. So what you have really is genomic and phenomic information. And then, you obviously need, for this type of study, you need an external validation data bank. Let's hold that for a second. And then I also have data from public repositories, whether it's genotype, phenotype, tissue association. And then I can also, if I do identify some significant signal, maybe I can go and test it in an in vitro testing or in vivo testing, actually. So this is in a way one of the workflow that we have leveraged here. So we started with a database that includes demographic data, disease diagnostic, lab test results, drug information, and all that kind of comes from the data warehouse. And then also more than 11,000 patients with their genomic information as well. So the question that we were interested in, that we wanted to understand if there is any loss of function variants. There are a few already that's been reported, but is there anything that we could find from our own? Because our biobank is a little bit unique on its own. So we want to understand, if you look into these few cardiometabolic traits, can we identify a few loss of function examples? So we have a lot of challenges with that, because we're really handling here with very messy type of data. There will be incorrect values, medication effects, and we used an approach called computational phenotyping. The long story short, we cleaned the data. The data is now ready for computing, and then we did a simple modeling approach, gene by trait association, as you can see in the equation. And then let's say if I'm interested in a loss of function variant in ApoC3, compared to a wild type variant, as you can see that the patients with the loss of function variant have a significantly reduced triglyceride effect. So that's a classic example. Everybody knows that, and it has implications in developing therapies and all. But then we looked at a variety of them. Like we indeed replicated some of the big ones, whether it's PCSK9 or ApoC3, but we also find something new as well. So we basically kind of integrated that data, like the kind of phenotype data, and then did some candidate association. And then what we identified was, we found a particular variant in a gene called DGAT2 could lead to, in a way, significant lowering in glucose levels. So we kind of followed that up with both, experimental studies. And then what happened was that we were able to find one gene, like especially based on these approach, with the validation that, so our mouse knockdown study suggests that DGAT2 as a potential target for glucose lowering therapy. And then we have identified a few others as well. Let's say if you're interested in lowering triglycerides, we found something, a few genes including RNMTL1, SCRN2, PCK2 as potential targets for lower cholesterol. So think about it. This is only a small database, and it's a fairly large EHR data, but like the linked data is very small. And UK Biobank has just released with hundreds of thousands of patient sample. So really, I want each and every one of the students in this call to kind of think about what's that most curious question that you want to ask. Data is never a problem. Like, data is always available. It's really how creative you can be, how curious you can be, and then you can ask these type of questions, and you can take it to either a therapy, a mechanism, or a target. So that's just one of my first example. And then the other potential theme I want to discuss, and this is again, so those who have heard about repositioning, this might be a little boring. For those who are hearing this for the first time, repositioning is an approach where we take an approved or a drug in a certain drug or a therapeutic in a certain stage of development. We take that drug, we investigate whether that drug could work for any other indication. And like you can see in literature, a ton of examples like that. And then what we can do is, like, if you could identify that using a drug that's in advanced stages of development, that could potentially be developed into a therapy for a new disease. So we could potentially save some development years, and then also the cost, time, and energy too. But at the same time, you still have to do repositioning is not just a computational part, but you have to do downstream analysis, downstream clinical trial. But the time to trial is where we are going to save, actually, rather than, it's not like when we hear about repositioning, don't think that it's an experimental approach where we do completely in silico. That's not the purpose or the aim of repositioning. You can do that, but really the value will come when you take it to a trial. And then there are a lot of different ways to do repositioning. And one approach that we've been focusing on, this type of a molecular matchmaking approach, using one type of data here. This is primarily transcriptomic data. So we can generate what we call as drug signatures and disease signature. And the idea is that, using statistical learning approaches, can we identify let's say if this is the disease signature for, let's say, disease 2, and I want to find a drug signature that could reverse these red to green, and maybe this is a good drug candidate for this particular disease. So can you do that type of a molecular matchmaking for that? And then we were interested in repositioning and did one of the largest meta-analysis in this space. And what we found, and I want each and every one of you to think about this and tell me your thoughts also. When we looked at around 200 or so, 200 plus repurposed agents across 1,100 diseases, what we found when we did a post hoc analysis, these repurposed, let's say I have drug disease 1 and disease 2, and that can be potentially targeted using drug X, they are more likely to have shared genetic architecture. For 31% of them, more than 31%, we saw that. And then again, around 27%, we saw that those two diseases, when you look into the pathways they share, we saw pathway crosstalks, we quantified them. And then the most interesting part, just like, by integrating the molecular data with EHR data, or clinical data, what we found was, if two diseases are comorbid, we see them most commonly within a patient population, they're more likely to potentially be repurposable. This is not a hard and fast rule for it, but again, there is some evidence that's coming in. These are the areas where you should potentially look when you're planning your repositioning investigation. And why repositioning works? Because one of the potential reason repositioning work is because it's pleiotropy. Pleiotropy is a key biological phenomenon, and where one gene could influence two or more seemingly unrelated phenotypic traits. And that might be the reason where, and then the genes are doing different functions in different settings. So when you put to using, one drug against two diseases, maybe there is a better targeting strategy there. So just for you to think about it. And as an example, so we recently, this is again a work that we've done at Mount Sinai. So here, what we did was in collaboration with my friend and colleague, Kamlesh Yadav, who's now at Texas A&M. And what we did was we're interested to come up with a therapeutic strategy for a rare form of prostate cancer. So we did some public data analysis, identified some gene expression data from GEO, and then we basically run a repositioning algorithm. We were able to identify, some of these drugs, and then what we found was, interestingly, an alkaloid, of course, that naturally long black pepper compound called piperlutamine was doing really good in some of our viability assays. And when we looked into the literature and we saw that this could be a potential target, because there's already been a ton of data on this in the literature. So we went ahead and built a mouse model for this unique sort of rare cancer type, and was able to show that the drug indeed have an effect as well. So really, this is again one of the example I wanted to share with you. And in a similar way, if you're passionate about a particular disease of interest, I would highly encourage to include repositioning as a potential strategy in your research. So the last part that what I want to cover today is, we live in, we have seen that we live in a world of data influx, and we have this challenge of we have more data is being generated, but there is a huge delay in inference from this data. What I meant by that is, let's say my friends and colleagues in microbiome or genomic sciences or or multi-omics group generating these different types of data. They know what their microbial signature is associated with the disease. They know their genetic variants. They know their transcriptomic signals. But and then they interpret them independently. They never put it together and look biology in a more connected way. But biology actually operated in this level. It's an integrated approach. But what can we do about it? And on top of that, along with omic data, we also have custom assay data. A company like AstraZeneca, for that matter, any other pharma company will have some custom assays, imaging data, phenotyping data, clinical data, you name it. And then instead of analyzing them independently, can we analyze them together? This is a question that's been, really been challenging for us, and just like the entire industry here. And when we started our group, one of the first group that reached out to us, our microbial sciences group, reached out to us and asked, hey, Shamir, can you and your team help anything with this? And I was really happy, because hey, there's going to be a ton of data on this. We can apply some deep learning approach. And then what I realized was, interestingly, the sample numbers are always very, very small. And then you have this classic, analytical challenge where you have small n and a large p. And then what happened? And then we did an extensive search and literature survey and identified that, why we cannot really use single-omic techniques and then, just make a bigger or better version of that. But they do not take, statistically into the, integration aspect. And then they do not find relationship between the omics. So what we realized was, maybe we need to make a wish list of these omics approaches, and then potentially come up with a way that we could, then use to, integrate, analyze, and interpret these data sets. So what we thought was, okay, let's write our wish list here. So we started with this sort of wish list, and what we found was there are 3 key ways that you can do omic data integration. You can do an early integration, you can do an intermediate integration, or you can do a late integration. But then what happens was that it doesn't matter what type of integration you choose. We also want to make sure that the data represented in each of these data, or omic or non-omic type, should be equivalent. One omic should not dominate. You can still weight your omics if you want, and they should be generalizable. It should not be only work for transcriptomic or NGS-based techniques, or it should only work for imaging-based techniques. And then we need to do variable selection on each block, and then in a way that we have to also do predictive modeling on top of that. And two of the key part was, you have to understand the relationship between different types of omics, and then also it should not be confined to only omic data, but other types of data should also be able to add. And then, we came up with this approach, We did an extensive literature survey. And then instead of developing something brand new, what we thought was we start with a pretty decent package and then add features that doesn't exist in it. So we basically start with something called Diablos. As you can see that we have some objective and subjective classification approach that we took here. And then, we identified certain algorithms that could go in there. And then with that, on top of that, we have built some additional layers as well. So these are the key features. We can do a lot of normalizing and scaling within omics world. You can do single omic multivariate modeling. You can do multi-omic multivariate modeling. We've also come up with a new strategy. Let's say you've done all these analyses, then you can actually do what we call as a block rank and identify the best features from that and best model and most stable model, and then you can go do downstream experimental validation as well. So this is, in a way, I don't have time to go through everything, but what I can do is, this is a sort of a broad overview. As you can see here, we have a lot of different types of omic data can be onboarded once you do pre-processing, and then you can do 2 types of analysis, or rather 3 types of analysis. You can do, if you don't know the class label, you don't have a class label, it's fine. You can do unsupervised exploration. If you understand what's your classes, you can do supervised exploration and go all the way to the modeling part. If you want to do cross data set prediction, you can build models, whether it's random forest or support vector neural network, you can do that. So, and then in addition to that, we have other models that we're building towards, understanding the gene sets and repositioning and all of that as well. So this is, in a way, what we want to do here really is to address the challenge in this biological data interpretation problem. So the features are interpretable. 3 things if you have to, take away from my talk. So interpretable features, relationship between omic types, it can be inferred, and it's scalable to add new omic types. We have so far integrated with 12 types of data, and we strongly believe that you can add more types of data as well. So this is a quick example, like this is a dummy data sets that we have put together. So you can see we have control seropositive and disease for a cohort. And then this is our initial model. We do model selection and tuning. Models get better, do better separation. The first one is using this one is using mono-omic data, and this is using multi-omic data. You can see that. And in addition to that, we have an array of novel visualization approaches. You can also understand the stability of these models as well here. And then this is what's available. We will be sharing this with the community very soon, and watch this space. And with that, I would like to come to the summary. I strongly believe, biomedical healthcare or data science it can actually optimize, accelerate, reduce the cost of development. As you've seen a lot of examples today, and I'm sure you're hearing a lot more examples throughout this symposium. And as data scientists, be ready to push the limit of your comfort zone. It's a team sport, right? Like we all need different types of idea to push the limits, because we don't know what we're going to find. And that's the joy of being a data scientist. And then the more creative you are, the good handle of subject matter expertise that you have, you can definitely bring in a lot more value. And then, definitely in biology, everything is connected. It's important to ask bigger and bolder questions. And one more thing, we are always hiring, always looking for collaborators, and will be, potentially looking for interns and apprenticeship positions and all, looking to fill all of that. So please be in touch. You can find me on LinkedIn or Twitter, and we'll be in touch if you would like to connect, and we'll be happy to stay in touch. And I would like to acknowledge my team from AstraZeneca, and for the other 2 examples, the team from Mount Sinai. And with that, I'll be happy to take any questions. Thank you.
Great. Well, thank you very much for this great talk. And a couple of questions. So first of all, a personal question. How did you personally end up at AstraZeneca? And can you briefly describe your path?
Oh, sure. Yeah. So, in a way, I wanted to be a software engineer when I was, from straight out of my high school. And thank you for asking that question. So, and then, but I took biology for my high school. So in India, I'm originally from India. In India, you cannot really go to computer science with a biology background. So you have to do some kind of an undergrad in non-biology background and then go to computer science. So, and then I went to an undergrad in physics. And then when I was doing physics, second year or third year of physics, I heard that there is this subject called bioinformatics that combines, computer science and medicine. I'm like, wow, that's fascinating. That's really, really exciting thing. And then I wanted to get into a master's in bioinformatics. So instead of going to computer science, I completed my master's in bioinformatics, and I did my grad school from India under Professor Sadamni, like National Center for Biological Sciences. And I worked primarily on building machine learning models to understand protein sequence structure and interactions. And then while I did that work, I wanted to kind of get experience with other type of data. And that prompted me to go to Mayo Clinic for my postdoc. And I was trained with this whole EHR-based data, genomic data. It was in the beginning of the whole sort of genomic medicine, precision medicine phase in the US. We were one of the first sites for a project called Electronic Medical Records in Genomics, like 12 years ago, and was part of that, participated in several clinical trials, and then eventually switched to industry, at certain point, been doing some consulting and eventually switched to industry. And I'm with AZ for now 3 years. The reason I joined AZ 3 years ago, AZ was starting the data science group, and we were like only maybe like, my including myself and my boss, we were like 3 members. Now we are around 60. And now the whole data science and AI is actually 150 plus in AstraZeneca. So we have grown a lot, and really, really excited to bring, to see the power of, data science and AI in drug discovery. So this, I still remember, in one of the early days of my master's in bioinformatics, I hear that we are going to see precision medicine. Every hospital will have a bioinformatics team, which is something I've seen happening right now. You can see that NGS is in every hospital. I mean, like in one way or the other, the physicians are using that in like one way or the other. So we're really there. I mean, like this is our moment, and yeah. So that's the long story in a short way. Thank you.
All right, great. Thank you for that. Okay, so we do have another question. You mentioned that you did your postdoc at Mayo Clinic, and how different is it working kind of in basic research as opposed to, clinical type information? I mean, how different is the thinking and, the need there in terms of omics data?
So definitely, like, compared to the basic science group where we primarily work with, genes, proteins, and computers. That's where we were working on. Here you have patient data too, but at the same time, you have to think about what is patient data. In the end, what you capture in an EHR data is nothing but, think of it like a large, long experiment, right? Like, you're measuring biomarkers, you're measuring imaging data, you're measuring other types, like, medication data. These are all different types of data that you measure from your patient cohort. That's being compiled into, not always a user-friendly database. You always need to do a lot of processing because there's a lot of human errors, because this will be either typed in or translated by an automated transcription engine. You have a lot of clinical notes that you can mine through. So there are a lot of that pre-processing is required. But in the end, the way that I thought about that was, this is pretty much like an experimental data, but it's coming from, a physician and patient encounter, and that's how I kind of thought through that. And now what we're seeing that when we were back at Mayo, the challenge was to put genomic data into electronic health record. We have achieved that. It's there and everybody's aware, and we still need more training. We still need, the accessibility is not global. We need to improve that. All of that is for true. But now the field of the experimental biology is moving at much faster pace. And as you can see that, like I mentioned, biology is now more digital biology, and we have that gap. They're generating more and more data, but we are not able to interpret them on time. So how do we do that? And that's where approaches like Omics Fold and these kind of strategies and needed. And again, we cannot do it by ourselves. We need collaborations. We need to work with the community, and I really appreciate this invitation to kind of share our research with you all. Yeah.
Great, thank you. Another question. So while recruiting for bioinformatic roles in AstraZeneca, is a PhD an advantage, or should you have only a master's degree?
We have, I can speak for the group that I am part of, like what we are data science and artificial intelligence. So we have a senior data scientist, principal data scientist, and then assistant director, a director, senior director, and then executive director, all the way up to the VP. So that's sort of our career level in data science. We don't have an absolute requirement for a PhD. So if you're very good at, in some of the technical skills or scientific skills, have a passion to work on solving the type of problems that are related to drug discovery, development, and repositioning, or even the entire enterprise of drug development, including clinical trials or patient recruitment. We hire both PhDs and masters. It's not just even we have undergrads too, like a very few undergrads too.
Okay, perfect. So